Hello and welcome everybody to my channel Omni Bio Entrance. Today we will be discussing the National Entrance Screening Test NEST 2019 Session 2 question paper. I will be discussing only the biology section. Coming to the first question, cladogram is a diagram used to represent the evolutionary relationship between group of organisms. The following cladogram shows relationship between some group of vertebrates. From the option provided, identify the features that denote MNOPQR in the correct order. So M will be vertebrae. M will be vertebrae and sharks have got that vertebrae. Then bony skeleton is for ray finned fish. Then comes amphibians which has got 4 limbs. So O will be 4 limbs. Amniotic egg is for crocodile and birds. And egg with shells is for birds, and hair is for rodents and primates. So R will be hair. So answer will be option 1. Next one. The biomass pyramid of 3 ecosystems are represented below. They have given primary producer, primary consumer and secondary consumer. The ecosystem most likely are. So if it is forest, so option 1 is deciduous forest. M is deciduous forest. In forest, the majority of biomass, that is over here, is tied up in the wood and is not available to most herbivores. So majority of the biomass in forest. So the primary producers are more in number when compared to the herbivores, that is primary consumers. So it is a deciduous forest ecosystem. Then next one they have told N is marine. The marine community produces an inverted pyramid of biomass. That is because the producers are unicellular algae which divide so rapidly that a small biomass can support a much larger biomass of herbivore. So primary producer in marine ecosystem is algae, and a small number of algae is enough to support a large number of herbivores, that is primary consumers. So the marine ecosystem is having pyramid of biomass inverted. So N is marine. Next, coming to most of the biomass in a grassland is found in the green plants and most of the energy flows through them. So most of the biomass over here is concentrated to the grasslands. Then they are eaten by the primary herbivores and then secondary consumers. So O is grassland. So option 1, deciduous forest, N marine, O grassland is correct. Then next one, trypsin is a digestive enzyme of serine protease group. It is considered an endopeptidase, that is, the cleavage occurs within the polypeptide chain rather than at the terminal amino acids located at the ends of the polypeptide. Trypsin cleaves peptide chains mainly at the carboxyl side of the amino acid lysine or arginine, except when either is followed by proline on the C-terminal side or preceded by aspartic acid on the N-terminal side. It is also known that only surface accessible lysine or arginine are cleaved by the enzyme. Otherwise cleavable amino acids K and or R, if buried inside, cannot be recognized and cleaved by the enzyme trypsin. A fragment of protein has the following sequence of amino acids that may lead to 2 different structures P1 and P2 from the same peptide. In P1, the peptide is linear, while in P2, the peptide is folded with the 26th amino acid lysine buried deep inside. So this is the sequence of the amino acid of the polypeptide chain, and following a trypsin digestion, the number of peptide fragments that will be formed for P1 and P2 respectively are. Answer is option 3, that is 666. How we got, I will show it in the next slide. So this is the amino acid sequence that I have rewritten. Now it cleaves on the carboxyl side. I have continued the enzyme polypeptide in the second line also. This line is a continuation of the first line. Okay, because the space was not sufficient, I had to do it like this. Then this is the N-terminal region. This is the carboxy terminal region. So what does trypsin do? It cleaves at the arginine, that is R, and K, that is lysine residues on the carboxy terminal side. So here there is one cleavage. This is the lysine one. Here also one cleavage, but it doesn't cleave when an arginine or a lysine is preceded by a aspartic acid, that is D. So here, even though arginine is present, it will not cleave there because arginine is preceded by D. But here it will cleave at this position, that is at this R, there will be cleavage. Then here it is lysine, so it will cleave there, and here also arginine is there, no other proline or aspartic acid is present, so it will cleave here. But here the lysine, that is it is followed by, lysine is followed by proline, so it will not cleave here even though lysine is present here. And here also cleavage won't be there because proline is present. So how many strands you are getting, fragments you are getting, 1, 2, 3, 4, 5, 6 fragments for linear polypeptide. Now if it is folded one, you will get the same thing because here at the 26th lysine is buried deep inside. Now 26th lysine was not cleaved in the linear polypeptide chain because of presence of proline, and by in the peptide chain also the lysines will not be cleaved because trypsin is inaccessible to one which is found at the interior of the protein fold. So this lysine is also not cleaved. So there also you will get 6 fragments, that is 1, 2, 3, 4, 5, 6 fragments. Next one. Mammalian sperm cells are highly specialized male gametes produced only for the function of fertilization. During spermatogenesis, these cells lose several subcellular entities and retain only those which are absolutely essential. Therefore, the mature sperm cells from humans will contain 55S ribosomes, mitochondria and microtubules. So option 4 is the answer. They don't contain 80S and 70S ribosome. 55S ribosome is present in human male sperm cells. Then mitochondria are present as well as microtubules. Now next one. If the ratio of A plus G by T plus C in one strand of DNA is 0.7, the same ratio in the complementary strand will be. So A plus G by C plus T is equal to C plus T by A plus G in the complementary strand. Suppose we take A plus G by C plus T as X by Y, C plus T by A plus G will be Y by X. So X by Y is equal to 0.7, Y by X will be reciprocal of that, that is 1 by 0.7, that is 1.42. So answer is option 1, 1.43. Now next one. The second half of the distal tubule and the cortical collecting tubules of kidneys are equipped with the principal and intercalated cell. Of these, the principal cell reabsorb Na plus and excrete K plus and are equipped with receptors for the hormone aldosterone. In primary hyperaldosteronism disease, that is more aldosterone is produced, there is an excess production of aldosterone which causes high blood pressure and K plus imbalance. Spironolactone, a diuretic drug, is generally used to treat this condition. Choose the correct option for the mode of action of spironolactone. Now there is excess production of aldosterone in hyperaldosteronism disease. So the drug, that is spironolactone, what it will do is it will inhibit aldosterone. And since aldosterone has effect on sodium reabsorption and K positive excretion, the inhibitory effect of this drug on aldosterone will affect the sodium reabsorption and potassium excretion. So answer will be 4. It inhibits effect of aldosterone on Na plus absorption and K plus excretion. Next one. In a plant, red flower color is dominant over white. Tall stem is dominant over dwarf and round seed shape is dominant over wrinkle. A plant that is heterozygous for all 3 characters are allowed to self-fertilize. What is the proportion of the offspring expected to be homozygous for red flower color, homozygous for tall stem and heterozygous for seed shape? Answer is 1 by 32. How we got, I have explained in the next. So they have given red flower is dominant over white, So we will represent red flower by capital R and white by small r. Then tall stem is dominant over dwarf. So tall stem will be capital T, dwarf will be small t. Round seed shape is dominant over wrinkled. So round seed shape we will represent as S and wrinkled we will represent as small s. Now they have crossed a heterozygous plant which is heterozygous for all the three characters with another heterozygous plant, and these are the number of gametes that you are going to get from one of the parents, and this is from the second parent. Now they have asked for homozygous red flower, homozygous tall stem and heterozygous seed shape, that is RR, TT, SS. How many are present? So this is the Punnett square for the the mating of the heterozygous individual with another heterozygous individual. Now since they have wanted capital R and capital T, there is no use of writing this, the small r cross with the small r, because you are not going to get capital R over there. It will be two small r's only, but we want capital R. Then over here also there is no use writing for, but I have written it. There is no use actually for writing the Punnett square for small t and capital T, because we want two capital T's, not one capital T and small t. So by the end of this Punnett square, if you write everything, you will see that there are 1 and 2 out of 64, 8 gametes. So 8 into 8 will be 64. So total there will be 64 offsprings. Out of them 2 will be capital R, small, capital T, capital S, small s. So 2 by 64, that is 1 by 32. Now next one. Which of the following statement about photosynthesis and respiration are correct? Both photosynthesis and respiration produce ATP. That is correct. Respiration is an exothermic process while photosynthesis is an endothermic one. That is also correct. Respiration and photosynthesis are linked in the mitochondria through oxaloacetate malate transformations. That is also correct. Respiration produces NADPH. No, respiration produces NADH and photosynthesis produces NADPH. So the fourth statement is wrong and option 1, 2 and 3 are correct. So option 1 is correct. Now next one. When antibodies are mixed with their corresponding antigen present on the surface of animal cell, erythrocytes or bacteria, they cross-link these objects to form visible clumps. This serological reaction is called agglutination and is very similar to precipitation reaction. Both reactions are specific because they depend on the specific antibody and antigen pair. Antigen involved in precipitation reaction are much smaller and more soluble than those involved in agglutination, making the latter reaction easily detectable. So agglutination reaction is easily detectable when compared to the precipitation reaction. Now, which of the following options is best suited to make the precipitation reaction more easily detectable? Now, why is precipitation reaction not easily detectable? Because antigen involved in precipitation reaction are much smaller and more soluble than those involved in agglutination reaction. So what we have to do, we should take some carriers which will facilitate the visualization. So large particle size carriers will facilitate the visualization of the antigen antibody reaction, and this include attaching soluble antigen to large inert carriers. Inert carriers in the sense they will not favor a antibody binding to the carriers. So we should choose the carrier in such a way that it is large, it is inert, that is it does not elicit an antibody reaction such as erythrocytes or latex beads. Now large particle size of these latex beads will facilitate the visualization of antigen antibody reaction. They have asked how precipitation reaction can be more easily detectable, because precipitation reactions are the antigen involved is very much smaller and more soluble. So it can be made detectable by using large inert carriers such as erythrocytes or lactate beads. Differential centrifugation is a technique used to separate cell components on the basis of size and density. The larger and denser component experience the greatest centrifugal force and sediment to form a pellet at the bottom of the tube, while the smaller, less dense components remain in the suspension above, a portion called the supernatant. Repeated centrifugation at progressively higher speeds will fractionate cell homogenates into their components. So if you take the cell homogenate, centrifuge at low speed, you will get pellet 1 and supernatant 1. Supernatant 1 you again take and centrifuge at medium speed, you will get pellet 2 and supernatant 2. Supernatant 2 you again take and centrifuge at high speed, you will get pellet 3 and supernatant 3. Supernatant 3 you again take and subject to very high speed centrifugation, you will get pellet 4. 4 researchers P, Q, R and S jointly performed the centrifugation procedure from human liver cells to study electron transport chain, protein synthesis, tubulin polymerization and cytochrome P450 respectively. Identify the pellet used by the researchers for their respective experiments. Now there are 4 researchers who are interested in studying electron transport chain, protein synthesis, tubulin polymerization and cytochrome 450 respectively. Now they have to choose which pellet they have to take from the human liver cell homogenate. So if you subject a cell homogenate to centrifugation, that is at low speed centrifugation, the more denser and larger particle will settle down, that is pellet 1. So it can be whole cells, nuclei or even cytoskeleton. Then again you take the supernatant 1 and subject to medium speed centrifugation, you will get a pellet having mitochondrion. Again if you take supernatant 2 and subject to high speed centrifugation, you will get a pellet containing microsomes and small vesicles. Again if you take supernatant 3 and centrifuge at very high speed centrifugation, your pellet will contain ribosomes, viruses, large macromolecules. So based on this, we have to see which pellet each researcher is going to take. So pellet 1, low speed centrifugation, I told you it will pellet whole cells, nuclei and cytoskeleton. So who will be interested in pellet 1? So the one who is interested in tubulin polymerization, that is R. So because tubulin microtubules are part of the cytoskeleton. So R will be taking pellet 1. Now pellet 2, medium speed centrifugation. So pellet 2, I told you will get mitochondrion. So one who is studying electron transport chain will be interested in pellet 2. So P will be pellet 2. Now pellet 3. Pellet 3, I told you you will get microsomes and small vesicles. So here it is cytochrome P450 and S is interested in studying cytochrome P450, so he will take pellet 3. Now pellet 4 will have ribosome, viruses and large macromolecules. So that will be by the one who is studying protein synthesis, because ribosomes are involved in protein synthesis. So pellet 4 will be taken by Q, who is interested in studying protein synthesis. Now, a sex-linked recessive gene in humans produces color-blind men when hemizygous, that is XCBY, and color-blind woman when homozygous. A non-linked autosomal but sex-influenced gene for patterned baldness is dominant in men and is only manifested in women when they are homozygous dominant. A heterozygous bald color-blind man marries a non-bald woman with normal vision whose father was non-bald and colorblind and whose mother was bald with normal vision. Choose the correct options for the phenotypic expectations for their children. I have given here all the options are correct, and how I have got them I have given in the next slides. So you have got a man, heterozygous, color-blind man. Now he is going to marry non-bald woman with normal vision. So we don't know the genotype of this non-bald woman with normal vision. Now the genotype of the woman we can find out using the father's genotype of the woman. So he is BBX, that is non-bald and colorblind, whereas mother of the woman is bald but not colorblind. So when we cross it, we will get this genotype of the woman, that is this one. Now when you cross the man and the woman, so this one crossed with this genotype, you will get this many progenies. After crossing, you will get 4 gametes from each of the crosses. Will be produced by man and woman, and upon crossing you will get 16 progenies. That is bald colorblind female 1, bald colorblind male 3 will be there, normal colorblind female 3, normal colorblind male 1, bald normal vision female 1, bald normal vision male 3, normal vision female 3, normal vision male 1. So out of a total female 8 and total male 8. So you can go through this one. Then next one. A purified linear double-stranded DNA fragment was independently subjected to 5 treatments using different restriction enzymes, and the unique fragment sizes of DNA thus obtained were measured using conventional agarose gel electrophoresis. The result of such treatment are depicted in the table below. No digestion, you will get 9 kb band. If you digest with EcoR1, you get 2 and 7, and if it is with HIN3, you will get 3 and 6. Enzyme HIN3 and EcoR1, you get 1, 2 and 6, and enzyme NDE1, you get 4 and 5. NDE1 and EcoR1, you get 2, 3 and 4 bands. Which of the following options is or are correct? Restriction enzyme site for NDE1 lies between those of HIN3 and EcoR1. That is wrong. Restriction enzymes, how I have explained in my later slides. Okay. Restriction enzyme site for EcoR1 and NDE are 3 kb apart. Restriction enzyme site for HIN3 lies between those of EcoR1 and NDE1. A double digestion with HIN3 NDE1 give rise to 2, 3 and 4 kb fragments. Now this is the agarose gel I have drawn for restriction digestion with EcoR1, HIN3 plus EcoR1, NDE1 and NDE1 plus EcoR1. These are the various bands they have given in the question. That I have just constructed the agarose or drawn the agarose gel roughly. I have marked the bands. Now when there is no digestion, you get a 9 kb base pair in the band. Now if you digest with EcoR1, you get a 2 kb fragment and a 1, 2, 3, 4, 5, 6, 7 kb fragment. If you digest with HIN3, you get 3 kb fragment and a 6 kb fragment. Now if you digest with EcoR1 and HIN3, you get 2 kb, 1 kb and 6 kb fragment. This is how, because EcoR1 will cut here, HIN3 will cut here, and you will get 3 fragments. Now if you digest with NDE1, it will cut over here and you will get a 4 kb and 5 kb band. Now NDE1 plus EcoR1, you will get now NDE1 plus EcoR1, you will get around 2 kb, 3 kb and 4 kb. Now NDE1 plus HIN3, we have to find out. We know HIN3 digests at around 3 and EcoR1 at 2. So you will get a 3 kb band, a 2 kb band and a 4 kb band. So option last 3 options are correct. Now, a transgenic crop plant was generated by transforming it with a kanamycin resistant gene P that integrates in its nuclear genome. Positive transformants were screened using this antibiotic as a selective agent. Southern blot analysis revealed a single copy insertion. Which of the following statement is correct? P bar minus transgenic line have a stronger expression level of gene P than the PP. No, the PP will have a stronger expression level than this one. If the transformants are self, the offsprings have either PP, P minus or minus minus genotypes. How I got that? I have put it in my next slide. Then Southern analysis band pattern for this and this will not be the same. Yes, you will get different band pattern. Then PP and P minus genotype plants should survive the selection. Why? Because they are positive transformants having kanamycin resistance gene P. So they will survive the selection. Now this is the cross between P and P minus and P minus, and you get the Punnett square, and you will get the plants with having PP genotype, P minus minus genotype, as well as minus minus genotype. Now next one. Approximately 400 million years ago, closely related fish species Q got separated from species P by continental drift. Species Q got confined in a small region where environmental temperature ranged between 4 degrees Celsius and 10 degrees Celsius. The species P got populated in a bigger region where the environmental temperature ranged between 24 degrees Celsius and 35 degrees Celsius. So species Q got confined to the colder regions, whereas species P got confined to somewhat warmer regions. So genome sequencing and other physiological analysis established the following facts. So species Q has high energy expenditure ratio, that is reproduction by muscle development. Yes, species Q reproduces throughout the year and it will be having high energy expenditure ratio. Then species Q achieved adaptive benefit by reducing its genome size. Yes, species Q have to live in cold conditions, so it has reduced its genome size to 15 billion base pairs, while species P it is 30 billion base pairs, and eliminating its redundant genes. So it has also eliminated some of its redundant genes also. So that was how it got its genome size reduced. Then species Q makes more ATP and tends to spend less time for reproductive purpose. It spends more breeding duration for species Q is throughout the year. So option 3 is wrong. The missing genes in species Q may provide clues about regulation of body size and longevity. Yes, because species Q got separated from species P by continental drift. So if we study the missing genes in species Q, then you can get an idea about regulation of body size and longevity. So options 1, 2 and 4 are correct. A group of researchers performed a genetic survey of 2 different human populations, one living in a deep tropical forest and another in a faraway metropolitan city of the same country. Their hemoglobin genes were analyzed and classified as normal or sickle cell anemic. 5 years later, the same group of researchers performed another survey for the occurrence of malaria in these 2 populations. The reports are tabulated as follows. So we can see from the graph that the number of individuals in the population in the forest is less than that in the city. The number of individuals with normal hemoglobin in the forest, it is less when from the forest population, whereas in the city only 40,219 are having normal hemoglobin gene. The number of individuals with sickle cell anemia is 30 in forest and 0.6 in the case of city. So city is having the number of individuals carrying sickle cell anemic gene lesser in number, and the forest is having more number of individuals. Now total number of individuals in the population. By 2015, the number increased both in forest and city. The number of individuals who had malaria in the last 5 years. In forest it was 3,516 and in city it was 4,113. Number of individuals died due to anemia in the last 5 years, that is only 35, and in city it is 355 individuals who died of anemia. Now, sickle cell anemia manifests in an individual because of a defective hemoglobin gene. Assuming that the rate of exposure of malarial parasite remains constant both in forest and in the city in the last 100 years and other compounding factors do not exist, which of the following statement is more likely correct? So, the percentage of individual dying due to anemia in both forest and city is almost the same. Yes. So, out of the total 4,215, 355 died, and that if you take the percentage of that, you will get 0.8. Similarly, in the forest, out of 4,455, 35 individual died of the anemia, and out of this if you take the percentage, you will get somewhere around 0.78. So the percentage of individuals dying due to anemia in both forest and city is almost the same. Then individuals carrying gene for sickle cell anemia are better protected against malaria. Yes, because the parasite, the malarial parasite will not be able to survive in the sickle cell gene. Sickle shaped hemoglobin. So, individuals carrying gene for sickle cell anemia are better protected against malaria. Then malarial infection is independent of the hemoglobin gene. Yes, that is correct. Individuals carrying gene for normal hemoglobin are better protected against malaria. No. Individuals carrying sickle cell gene are better protected. So that's it. Thank you. Please like, share and subscribe.